So last lecture, we talked about the beginnings of cell respiration, and mainly we focused on glycolysis, the first step, which is universal to all organisms, and then we talked about fermentation, which is where after glycolysis, no oxygen is present, and so we also call that pathway anaerobic respiration, the glycolysis to fermentation pathway. So today we're going to focus on the other option. If we go through glycolysis and oxygen is present, what happens next? So the remainder of cell respiration is going to occur in the mitochondria. Now you may recall that the first step, glycolysis, occurred in the cytoplasm, and fermentation also occurs in the cytoplasm. So if oxygen is present, the remainder of cell respiration will happen in the mitochondria, which we know is the powerhouse of the cell. So the mitochondria has two membranes. The inner membrane has these specific folds called cristae, and these are actually going to be very important, so I'm going to highlight them here. So these are the cristae, where it folds in here, 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 all of these are your cristae. And this is going to be very important, specifically, I'll add it here, for the ETC, the electron transport chain. And then the center here is called the matrix, and you can see it's filled with fluid. Now, there's also a space in between the two membranes called the intermembrane space, inter meaning between. So you have the inner membrane, which contains the cristae, folds, the outer membrane here, and then the space between them, which is the intermembrane space. And all three of those are actually going to be important in the remainder of cell respiration. So, now, there technically are three steps in cell respiration we talked about. Glycolysis, this is for aerobic, glycolysis, the Krebs cycle, and then the ETC, the electron transport chain. However, there is another step, and it's in between glycolysis, at the end of glycolysis and the beginning of the Krebs cycle. And that step is called the oxidation of pyruvate. So pyruvate, if you remember, this is pyruvate. It is a molecule that only has 3 carbons, and it was a product of glycolysis. That was what was made when glucose got split in half. So technically at this point there would be 2 of those because glucose broke in half, and this is the cytoplasm. So it turns out before pyruvate can start the Krebs cycle, which is going to look kind of like a circle, it's going to be a cycle with several steps, it has to go through a little conversion. And this conversion happens, as you can see, as it's entering the mitochondria. So what it does, it gets converted to another molecule called acetyl-CoA or acetyl-coenzyme A. Ultimately what it is that the pyruvate loses one of its carbons, and you can see that's released as carbon dioxide. Another NADH is made. If you recall, there was this coenzyme called NAD+, and it becomes NADH. And then this coenzyme called coenzyme A, which they're not really drawing, it's kind of big and bulky. It hooks to what's left here, which is called an acetyl group, and so the whole thing is called acetyl-coenzyme A. You don't have to know the drawings of what these look like, but this step happens in between. The reason it's called oxidation of pyruvate, and I don't know if you paid attention to the little Amoeba Sisters video we watched yesterday, but pyruvate, it becomes what's called oxidized. Oxidation is when it loses an electron. So because pyruvate loses an electron in this process, it is becoming oxidized, and so it's called oxidation of pyruvate. 2 carbon dioxides are released. You might say, but wait, in the picture I only see 1. But remember, there are 2 pyruvates, so there's 2 carbon dioxides. There's 2 NADHs, and there's 2 acetyl-coenzyme A's that are made. So that is why it's 2, 2, 2, not 1. It's 1 per pyruvate, but we had 2 of these at the end of glycolysis because glucose split into 2. Alright, and so this step is called the oxidation of pyruvate. It's kind of a minor step. It's not counted as one of the 3 major steps, but it does have to happen so that the pyruvate is now turned into something that's ready to go into our next major step, which is called the Krebs cycle. This is just another picture showing the oxidation of pyruvate. So pyruvate has 3 carbons. They're just drawing the carbons here. And this is showing it now becoming, it only has 2 carbons. The third one became CO2, hooked up with some oxygen there. And then NAD becomes NADH. Also to clarify, the oxygen here is actually coming from the pyruvate. It's not the oxygen we breathe. The oxygen we breathe does not come into this process until the very, very, very end. So pyruvate itself provides the oxygen and carbon that become the CO2. All right, so the Krebs cycle has another name. It's also called the citric acid cycle. That's because citric acid or citrate is the first thing that's formed, and it is a cycle that like goes in a circle. You do not have to know all the steps. You should know where it occurs, which is in the mitochondrial matrix. Remember, that was the center part here of the mitochondria, the matrix. What it does, it takes the pyruvate, which technically is not really pyruvate anymore. I probably should have clarified. This is actually the acetyl coenzyme A, because the pyruvate has gone through that step, so it's not really called pyruvate. But it's going to convert that into carbon dioxide. It's also going to make some more NADH, and it's going to make another molecule called FADH2. And that comes from FAD. So NAD was a coenzyme that could pick up hydrogens, and FAD is a different one that can also pick up hydrogens. And this also makes 2 more ATP. Now, I don't know if you remember, but glycolysis made a net of 2. So now we're up to 4 ATP that we've made for glucose. So at this point, at the end of this cycle, all of the glucose that entered, we had a C6H12O6. All of the glucose has been converted into carbon dioxide. All we're left with now are the hydrogens. All of the hydrogens from glucose are still going to be continuing through to our next step. But the glucose itself has all been turned into carbon dioxide, and that carbon dioxide is a waste product that we exhale. So the carbon and the oxygen from glucose, gone. They are now CO2, we exhale them, and the only thing left is the hydrogen. And if you think about it, that must mean the hydrogen is very important, because at this point, we only made 2 ATP net in glycolysis. We made 2 ATP in the Krebs cycle, so we've only made 4, but if you go back to the original equation, we're going to end up making a total at the end of approximately 36. Some books will say 38. So obviously, this last step that's coming must be a really big step, because it's going to make almost all the ATP. Here's a picture of the citric acid or Krebs cycle. Notice it makes carbon dioxide, it makes a couple of ATP, it makes some NADH and some FADH, and really it's just making those from NAD and FAD. It's not making them from scratch, it's just these are picking up hydrogens. The reason this says 6 is they're combining, they are actually combining the oxidation of pyruvate with the citric acid cycle. Technically, the citric acid cycle itself only makes 4 CO2s, and the other 2 originally were from the oxidation of pyruvate step, that little step before. So right here. So here's the step, that the oxidation of pyruvate step. This is the acetyl-CoA from that step entering the Krebs cycle or citric acid cycle. Do you have to know these steps? No. But I would just like you to note that, see what it's doing? It's breaking down the glucose the rest of the way. At the end of this cycle, this last molecule ends up going back in. So it's almost like a Ferris wheel, where this gets on and it goes through the process, and CO2 gets off, and then the process starts all over again. So it's kind of like a little Ferris wheel, where people are getting on, people are getting off, and then the seats kind of stay. So this molecule stays, becomes this, gets back to this again. Also notice here's the NADH is being made, the FADH is being made, here's ATP being made, so it makes a bunch of things. All you, at the end of the day, need to know about it is that it makes more NAD and FAD, which are important. You don't have to know how many, you're not going to be tested on the numbers. It makes a couple of ATP, 2 total, per glucose. That starts at the very, very beginning of glycolysis. Total of 2 are made in this citric acid cycle. It turns 2 times, makes 1 ATP on each turn. And it also makes some CO2. So by the end of this process, all the glucose has been converted to CO2, and you exhale that. That's not really useful for you. And that brings us to our very last step, the ETC, or electron transport chain. So it happens on those inner folds of the mitochondria called the cristae. the NADH and the FADH2, the two coenzymes, are now going to release all their hydrogens. And they're going to go back to being NAD plus again, which can then go back and start all over, and FAD, and that can go back and start all over. And these hydrogens are going to basically be part of something called an electron transport chain. This is going to generate power, which I will show you. But basically what happens is these dump off their electrons. The electron and a hydrogen are not the same thing. The hydrogen and the electron separate from each other. So you end up with a hydrogen ion with a positive charge and then an electron that was part of the hydrogen. The electron goes down what's called an electron transport chain. I'm going to show you some animations of it in class. But it basically is just, it's sort of like playing hot potato. It's getting passed from protein to protein to protein in the inner cristae. At the end of the electron transport chain, it's going to get picked up by oxygen. Oxygen is waiting at the end. This is where finally we know why we have to breathe the oxygen. It's waiting at the end of the electron transport chain. When the electrons go through this electron transport chain, that generates power. It's like electricity. What that power is used to do is to pump the hydrogen ions into the intermembrane space. Remember, that's the space in between. So this is happening in this membrane, and all the hydrogen ions, without their electron, are getting pumped into this space here. And they're sort of trapped there temporarily. That takes energy. So the only way they can get back to the middle and hook up with oxygen, which will ultimately make water, the only way they can get back there is through a special enzyme called ATP synthase. And as they flow through ATP synthase, that is what's going to generate ATP. And so this process makes an additional 32 to 34 ATP. And it kind of depends on the organism. There's a couple of like mini things that happen there. And that's why the number can be different. So at the end of cell respiration, they usually say that you make about 36 ATP. Some books will say 38 ATP. You only made 4 between glycolysis and the Krebs cycle. And then all the rest are made here in the electron transport chain. So here's a picture of it. So here's the NADH dumping off its hydrogen. This is the FADH also dumping off its hydrogen. The electrons only, which remember is part of an atom, they get passed from protein to protein to protein. Do you see that happening? And the electrons come out here. So this is the electrons, the little negative charges coming out here. Notice this generates power, and the hydrogen ion, without its electron, the H+, gets pumped into this space, the intermembrane space between the two membranes. This is a mitochondria, not a cell. So we're talking about this space. This is the inner membrane. This is the outer mitochondrial membrane, and this is the space here. These all build up, and their only way to get back in is to go through ATP synthase. And when they go through this enzyme, boom, ATP is made. And this is sometimes called chemiosmotic or oxidative phosphorylation because it involves electron transport and something called redox reactions, which we don't really talk about in here. But ultimately, it's a way of making ATP that's much more efficient than glycolysis in the Krebs cycle. It makes a lot of ATP. Here's the whole thing put together. So you've got glycolysis, oxidation of pyruvate, Krebs cycle or citric acid cycle, electron transport chain. Notice 2 ATP net, 2 ATP, another 34. So that adds up in this scenario to 38. But again, depending on what book you look at, some books are going to say 38, some are going to say 36. Here's just anaerobic versus aerobic. So they all start with glycolysis, but anaerobic would be this whole pathway going to the left, and then aerobic would be this whole pathway going to the right. So they would share glycolysis. And a little bit about exercise. So aerobic exercise allows you to make ATP with oxygen. So for example, marathon running. You're using oxygen. Your heart rate kind of goes up a little. Soccer and basketball are about 80% aerobic. Anaerobic is when you run out of ATP and you don't have enough oxygen, like sprinting, heavy weightlifting. The problem with that is that you do build up lactic acid and you can't maintain it for a long period of time. We're going to do a lab with exercise. And then once oxygen is present again, you would go back and restore those levels. So respiration itself is breathing. It is related because you increase your respiration when you need more oxygen for more cell respiration. You make more CO2 as well. And that's it. Photosynthesis and respiration are connected. The products of one are the reactants of the other.